I want you to try something right now. Just reach out and pick up a coffee cup. Or, I mean, if you're walking, just focus on taking your next breath.
Right.
Every single visible movement you just made, like everything you do to interact with the physical world, it relies on this completely invisible, microscopic tug of war that's happening inside your body millions of times a second.
It really is a paradox, honestly, because we take movement completely for granted. It feels so seamless to us.
Yeah, you don't even think about it.
Exactly. But the biological machinery operating under the surface to make that movement happen is an absolute marvel of molecular engineering.
And that actually brings us to our mission for today's deep dive. We are looking at this fascinating stack of anatomy and physiology sources to explore something called the sliding filament theory.
Which is such a great topic.
It is. Essentially, we're going to look at the fundamental step-by-step mechanism of how a muscle actually contracts. So, okay, let's unpack this, because before we can understand how muscles actually work, we have to look at how scientists, thought they worked for a really long time.
Right. For decades, the dominant model in biology pictured muscle filaments behaving sort of like a coiled spring.
Like a literal spring.
Yeah, or like a protein cable that could physically fold in on itself.
So if I have like a piece of paper in my hand,
Right.
I crumple it up into a tight ball. The paper itself is taking up less physical space end to end. It actually shrank.
That was the exact assumption. They thought contraction meant the actual physical filaments inside your muscles were shrinking or coiling up. They were drawing loosely on how other structural proteins behave, like keratin.
Right.
And this folding model actually had some support from early X-ray diffraction work, but it had a massive fatal flaw.
A flaw that got exposed by this incredible historical accident in 1954, right? The sources call it the Great Huxley Coincidence, and it is just wild.
It's one of my favorite stories in science history.
Right, so in the May 22, 1954 issue of the journal Nature, there were 2 back-to-back papers published that completely rewrote our entire understanding of muscle movement. And they both concluded that muscle filaments do not shrink at all.
But the real coincidence is the authors.
Yeah.
One team was led by a scientist named Andrew F. Huxley. The other team was led by Hugh E. Huxley.
They shared a last name.
They shared a last name. They were working on the exact same highly specific biological problem. They published in the exact same journal on the exact same day.
But they were completely unrelated.
Completely unrelated. They had never even collaborated.
So how did they arrive at the exact same conclusion without working together at all?
Well, what's fascinating here is how their completely different experimental approaches converge to prove this undeniable geometric truth.
Okay.
Andrew Huxley's team, they used interference microscopy to look at living frog muscle fiber.
Living tissue, right?
Meanwhile, Hugh Huxley's team used electron microscopy and X-ray diffraction on fixed, dead muscle tissue. So 2 totally different imaging techniques.
But they saw the same thing.
Yes, they both observed the same structural geometry inside the muscle's microscopic functional unit.
Which is called the sarcomere.
Exactly, the sarcomere.
So they were looking at the banding patterns, right? Like
Yeah.
the dark bands and the light bands you can see under a really powerful microscope.
Correct. They saw that the darkest band, which is the region containing the thick filaments, it stayed a perfectly constant width when the muscle contracted.
Okay, so the dark part doesn't change.
Right. Meanwhile, the lighter bands and the little gaps in the middle, those shrank.
So if the thick part isn't changing size, but the gaps around it are getting smaller, I mean, the thick filaments themselves literally cannot be shrinking.
The only way that geometric pattern is physically possible is if 2 separate, constant-length filament systems are sliding past one another.
Wow.
The filaments aren't changing length, only the amount they overlap is changing.
So going back to my paper analogy. If the old model was crumpling up a piece of paper, the new model is like taking your 2 hands, interlacing your fingers, and sliding them together.
That is a perfect visual analogy.
Your fingers don't get shorter, they just overlap more.
Yes. And that reframing, going from proteins coiling up to structures sliding past each other, it was a massive paradigm shift.
Oh, good.
But of course, that 1954 theory was purely structural. It proved that they slide. It did not explain how they slide.
And figuring out the how actually took scientists another 15 years.
It did.
So let's zoom in on that molecular tug of war. How does an electrical signal from your brain actually translate into physical sliding inside that microscopic sarcomere?
Well, first, we need to appreciate the scale we're talking about. A relaxed human sarcomere is roughly 2.0 to 2.2 micrometers long.
Which is tiny.
It's about 1/50th the width of a human hair.
Oh, wow.
Yeah. And inside this tiny unit, you have those thick filaments, which are made of a motor protein called myosin. They sit right in the center.
Okay, myosin in the middle.
And then you have thin filaments made mostly of a protein called actin, and those are anchored to the outer edges.
Right. So the brain decides to move a muscle, and a nerve sends an electrical signal. But, like, electricity can't physically grab a protein and pull it. What bridges that gap?
Chemistry.
Ah.
The motor neuron releases a chemical transmitter called acetylcholine, which triggers a wave of electricity that travels deep, deep into the muscle fiber.
Okay.
And that electrical wave hits an internal storage tank inside the cell called the sarcoplasmic reticulum.
The sarcoplasmic reticulum.
Yeah.
And what's inside that tank?
Calcium. The electricity basically forces the tank to dump a massive flood of calcium ions right into the cellular fluid.
Now, here's where it gets really interesting, because a really common misconception I noticed in the reading is that people think this calcium binds directly to the thick myosin filament to, like, activate it.
Right.
But that's not what happens at all, is it?
No, not at all. The calcium actually acts as a key to unlock the thin filament, the actin.
Okay, how so?
Because in a resting muscle, the binding sites on the actin filament are physically covered up. There's a protective protein called tropomyosin just sitting over them.
Ah, so it's blocking the connection.
Exactly. The myosin desperately wants to grab the actin, but it's physically blocked.
It's like having those little plastic safety caps on electrical outlets so kids don't touch them.
Very much so. So the calcium floods in and binds to a different regulatory protein called troponin.
Okay.
And when that happens, the troponin physically shifts the tropomyosin out of the way.
The safety caps are pulled off.
Yes, safety caps are off. So now the binding sites on the actin are totally exposed. And the thick myosin filament has these little heads sticking out of it, and they finally have something to grab onto.
So they reach up and physically connect.
Which brings us to the main event, the power stroke.
The actual pull.
Right. Now, the myosin head is already locked and loaded. It's holding onto a molecule of ADP and a phosphate ion from a previous chemical reaction.
Just waiting.
Just waiting. And when the myosin binds to the actin, it releases that phosphate ion. That release acts like a chemical trigger.
Okay.
The myosin head pivots on its hinge, dragging the actin filament roughly 5 to 10 nanometers toward the center of the sarcomere.
Wow. And then it releases the ADP.
Exactly. It releases the ADP.
Okay, now I had to stop and reread this next part in the sources because it really threw me.
The ATP part.
Yes. Because after the pull, the myosin obviously has to let go so it can reach out and grab the next spot.
Mm-hmm.
And the sources say it does that by binding to a fresh molecule of ATP, which is the body's main energy currency.
Right.
Wait, if ATP is energy, wouldn't it be used to fuel the actual pulling motion? Like, why is the body burning its precious energy just to let go?
It is incredibly counterintuitive. You would naturally think the energy fuels the contraction itself.
Right.
But you have to think of the myosin head like a mousetrap.
Okay.
The energy required to snap the power stroke that was already stored in the spring from when you said it previously.
Ah. So the release of the phosphate just triggers the snap.
Exactly. The new ATP is actually required to pry the trap back open, resetting it. It provides the energy for detachment.
That fundamentally flips how I thought about muscle energy.
It does, and it also explains a very famous, somewhat morbid biological phenomenon.
Rigor mortis.
Rigor mortis, yes.
The stiffness of a body after death.
Exactly. Because after death, the circulatory system obviously stops supplying fresh ATP.
Right.
And without ATP, those myosin heads simply cannot detach from the actin. They remain rigidly bound.
Wow.
So, counterintuitively, the absence of ATP is what causes the muscles to lock into a contracted state. It's the lack of energy that causes the stiffness, not an active contraction.
That is wild. It's just locked there.
Yep.
Okay, so assuming you are alive and have a steady supply of ATP, the myosin head binds the ATP, it lets go, and it breaks that ATP down.
Right, into ADP and phosphate.
And that provides the energy to spring back into its cocked position, ready to bind and pull all over again.
You got it.
But it's important to visualize this properly, I think. Because it is not one giant synchronized heave where all the myosin heads pull at the exact same millisecond.
Oh, not at all. Across the thousands of myosin heads on a single filament, it is entirely asynchronous.
The sources have this great analogy for this. They say it's like a giant team of people pulling a really heavy rope hand over hand.
Yes.
Like everyone is grabbing, pulling, letting go, and reaching forward at completely different times. That way, the rope never actually slips backward.
Yeah, and that hand-over-hand pulling is happening across hundreds of filaments per sarcomere and thousands of sarcomeres in a single fiber.
Which actually brings up a huge question for me. We're talking about tiny protein heads pivoting 5 to 10 nanometers. A nanometer is a billionth of a meter.
It's unfathomably small.
Right. So how do we actually have the concrete evidence for this? How do we measure something that small?
Well, the evidence evolved over decades as the technology finally caught up with the theory. A major leap happened in 1966 with a classic study on what's called the length-tension relationship.
Okay. Who did that one?
That was Andrew Huxley again, working with Gordon and Julian. They took single frog muscle fibers and basically measured exactly how much isometric tension they could produce at various stretched lengths.
And what did they find?
They found this microscopic Goldilocks zone.
Goldilocks zone.
Yeah, because if you stretch a sarcomere way too far, the thick and thin filaments are pulled so far apart they barely even overlap.
Oh, I see.
So because the myosin heads can't physically reach the actin, very little force is produced.
It's like trying to pull a rope, but only your fingertips can barely brush against it.
Precisely. You have no grip. But on the flip side, if you squish or compress the sarcomere too much before it contracts, the thin filaments from opposite ends start crashing into each other in the middle.
They get in their own way.
They interfere with each other, and again, the force drops dramatically, so the maximum tension only happens right around that 2.0 to 2.2 micrometer length, where the overlap geometry is absolutely perfect.
So force generation relies entirely on that perfect geometric overlap.
Exactly.
But that's still looking at the whole fiber, right? How did scientists eventually prove that these tiny individual myosin heads were doing the pulling?
For that, we have to fast forward to 1994.
Okay.
A team of scientists, Feiner, Simmons, and Sputich, they used an incredibly advanced piece of technology called a laser optical trap.
Wait, a laser trap? How do you hold a protein with a laser?
I know it sounds completely like science fiction, but you can actually use the momentum of tightly focused laser light to create a microscopic pressure field.
Really?
Yeah, it acts like a tiny tractor beam. So these scientists attached a single actin filament to 2 microscopic glass beads, and then they used the lasers to hold those beads suspended in fluid.
Wow.
And then they carefully lowered that actin down onto a single isolated myosin molecule.
Just one single molecule.
Just one.
That is insane.
If we connect this to the bigger picture, it represented a monumental shift in biology.
I mean, yeah.
Muscle physiology moved from calculating statistical averages of whole chunks of tissue to literal single-molecule mechanical engineering. Through that laser trap, they could measure the exact physical displacement of a single power stroke. That's how we know a single pull moves the filament roughly 5 to 11 nanometers.
And what about the force?
It generates a force of just a few piconewtons.
Piconewton, I mean, that is almost immeasurably weak. And yet, if I go to the gym and deadlift 200 pounds, I am just summing up billions and billions of these microscopic piconewton strength pulls all happening simultaneously.
It is the ultimate example of strength in numbers.
That is just incredible to think about.
It truly is. But, it's worth noting that the scientific consensus wasn't always quite so neat.
Right.
As technology improved, the sliding filament theory actually had to evolve. It wasn't the complete picture back in the 50s.
Right. The sources point out this really fierce debate about the mechanochemistry. What exactly were scientists disagreeing on there?
The debate was over how the myosin head actually physically moves. Hugh Huxley proposed a swinging lever arm model in 1969. He suggested the protein had a physical hinge that swung back and forth, kind of like a rigid lever.
Makes sense based on what we know now.
But other scientists at the time argued for a thermal ratchet model.
What on earth is a thermal ratchet?
Well, the thermal ratchet idea basically argued against a rigid hinge. Instead, it proposed that ambient heat causes the myosin protein to just jiggle around randomly.
It's just ambient body heat.
Yeah, and as it jiggles, it occasionally bumps forward into a favorable chemical position on the actin and just locks in the place.
Oh, I see.
So it essentially ratchets forward through random thermal motion rather than a coordinated mechanical lever swing.
But the swinging lever arm won out in the end, right?
It did, yeah. In the 1990s, researchers like Ivan Raymond used X-ray crystallography to map the atomic structure of the myosin head, and they finally proved that the physical lever-like neck domain, the actual hinge, it really did exist.
Okay, so they found the hinge, but then there is the discovery of titin.
Ah, titin.
I found this part fascinating in the reading, because we've got the thick myosin filaments and the thin actin filaments.
Yeah.
But in the late 1970s, they discovered a completely new third filament.
Yes, titin. And it is an absolute behemoth.
Huge protein, right?
It is the largest known protein in the human body. It spans half the length of an entire sarcomere, anchoring the thick filament to the outer edge.
So, wait, does the discovery of titin mean the original 1954 sliding filament theory was actually wrong?
Well, it doesn't mean it was wrong exactly, but it means it was structurally incomplete.
Okay.
Titin basically acts like a massive passive molecular spring.
A spring again.
Right, but a real one this time. It helps keep the thick filaments perfectly centered in the sarcomere, and it provides passive elastic tension, which stops the muscle from overstretching.
Oh, I see.
The original 1954 model didn't account for this passive elastic recoil at all. It just assumed a pure sliding system with two filaments.
So it's like they figured out how the engine of a car works, but they didn't realize the car also had this highly complex suspension system holding it all together.
That's a great way to contextualize it, actually. And honestly, we shouldn't forget the historical timeline here either.
How do you mean?
Well, long before the 1954 structural discovery, thermodynamic pioneers in the early 20th century knew something active was happening.
Right.
A.V. Hill won a Nobel Prize in 1922 for measuring the heat produced during muscle contraction. And then Wallace Fenn in 1923 showed that muscles produce extra heat when they're shortening against a physical load.
The Fenn effect. So they proved that muscles actively consume chemical energy to do mechanical work.
Exactly. They laid the thermodynamic groundwork decades before anyone even knew what a sliding filament was.
That's so cool. So what does this all mean for us today? Because we zoomed all the way down to individual atoms and piconewtons and laser traps. But why does understanding this exact step-by-step microscopic cycle actually matter in the real world?
Because this raises an important question about the fragility of our biology.
Okay.
Every single step of this contraction cycle is a potential target for modern medicine, or for extreme danger.
Danger how?
Well, by understanding the precise sequence of events, we can intervene. Take anesthesia, for example. Drugs like rocuronium are used during surgery to induce controlled muscle paralysis.
Right. You definitely don't want to be moving during surgery. And how do they achieve that mechanically?
They target step 1. Rocuronium blocks the acetylcholine receptors at the neuromuscular junction.
So the chemical signal from the nerve is just blocked.
Exactly. The electrical wave never starts, the calcium is never dumped into the cell, and the muscle cannot contract.
It just cuts the telegraph wire right at the source.
Exactly. But the sources also mention toxins and nerve agents doing the exact opposite.
Oh, right, like organophosphate insecticides.
Right. Instead of blocking the initial signal, they block the enzyme that normally clears the acetylcholine away after a nerve fires, so the muscle gets a continuous, non-stop contract signal. This causes uncontrolled excitation-contraction coupling, which leads to dangerous, sustained muscle tetany.
And tetany just means the muscle is locked in a permanent, violent cramp.
Precisely. It can be fatal if it hits the respiratory muscles.
Wow.
And this microscopic mechanism is also relevant to everyday medicine. Statins, for instance, are widely used to lower cholesterol, but in some patients, they cause myopathy.
Myopathy being the clinical term for generalized muscle pain and weakness.
Yes. Researchers are actively studying how these cholesterol drugs might unintentionally disrupt normal muscle cell function right down at the microscopic level of the sarcomere.
It even impacts space travel.
Yeah.
I saw in the sources they discuss how astronauts lose muscle mass in microgravity.
Right, because of the unloading.
Right, and it's entirely because these sarcomeres experience reduced mechanical loading.
The whole sliding filament system is incredibly dynamic. It actively responds to the physical loads placed upon it.
Use it or lose it.
Literally. If you don't load it, the body breaks it down to save energy. And when the structural proteins fail entirely due to genetics, the results are devastating. Think of Duchenne muscular dystrophy.
The genetic disease, yeah.
It's caused by a missing structural protein called dystrophin. Now, the sliding filaments inside the cell might be generating force perfectly well, but dystrophin's job is to anchor that pulling force to the cell membrane.
Ah, I see.
Without it, the force isn't transmitted properly to the skeleton, and the muscle cell essentially tears itself apart from the inside.
So the engine is running perfectly, but the drive belt is missing. The force never reaches the wheels.
That is exactly the mechanical failure point, and that's why understanding this deep physiology is so vital. It is the absolute foundation for the advanced gene therapies being developed right now in the 2020s that aim to fix these exact structural
Because you
deficits.
can't fix it if you don't know how it works.
You simply cannot fix the machine if you don't know exactly how all the microscopic parts move.
Okay, let's wrap this up. We started with the idea of crumpling paper, but now we know the truth. Filaments don't shrink, they slide.
They slide.
The dark bands stay the exact same width, while everything else gets pulled closer together. We learned the incredibly counterintuitive fact that ATP is actually needed to release the muscle, acting like the energy to pry open a mousetrap, which perfectly explains why rigor mortis sets in without it.
Right.
And we learned that massive human strength is really just billions of tiny myosin heads pulling hand over hand, perfectly choreographed by waves of calcium.
It is a phenomenal biological system. But I think the most important thing for you to take away from this deep dive is that the book is still not closed.
Really?
Yes. Despite knowing the basic sliding geometry since 1954, we are still figuring out how our own bodies move.
Even with the lasers and the atomic microscopes.
Absolutely. The sources note that Titan's role in something called active force enhancement is still being actively researched today.
What does that mean?
It turns out Titan might not just be a simple passive spring. It might dynamically change its stiffness depending on calcium levels.
Oh.
And with modern cryo-electron microscopy, scientists are still finding brand new conformational states of the myosin head at near atomic resolution. The picture is still getting clearer.
So the science isn't static, it's sliding forward too.
I see what you did there.
So the next time you casually reach out to lift that cup of coffee, just take a second. Consider that you are operating a piece of biological nanotechnology so complex, so perfectly calibrated, that the brightest minds and the most advanced microscopes in the world are still trying to map all its moving parts. Drink up.